Okay, now that we're done talking about the life cycle and how blood cells are born and how they die, and remember we have three types of formed elements. These are the cells of your blood. So we have erythrocytes, leukocytes, and thrombocytes, or red blood cells, white blood cells, and platelets. Now that we're done with the life and death of red blood cells, let's talk about white blood cells. Now, later on in the semester, when we talk about the immune system, we will talk about white blood cells more in detail. But for this upcoming exam you have in the next few weeks, that will be just focus on what's covered in the blood chapter and the slides so far, because it is easy to go down the rabbit hole when we talk about white blood cells. But what are the white blood cells? Okay, so we know there are white blood cells, but what do they do? Well, what they do is protect your body against disease and foreign pathogens. They are your own little personal army that you and little soldiers that help to defend your body against things that can cause disease. And pathogen is any foreign agent that can cause disease. And it's not just pathogens. Your immune cells are also important in protecting yourself against your own rogue cells. And what I'm talking about is cancer. So it's very important in like maintaining your overall health by protecting against diseases from either from foreign or from like things like cancer. Alright, so there are five main types of white blood cells, and they're classified by so what we see is like they can be classified by how they appear. And what we see are neutrophils, eosinophils, basophils, then monocytes, and then leukocytes, or lymphocytes actually. Lymphocytes, yeah, don't get leukocyte and lymphocytes confused. Lymphocytes are a type of leukocytes. All of these are leukocytes, but lymphocytes is a specific type. Now, some of you might be like, well, they don't look very white to me. In fact, one of them looks pretty red and pink, and this is because they're not actually, this is a false coloration. They've actually been fixed onto a slide and then stained with special chemicals to make them appear these colors. And white blood cells are not naturally pink or purple. If we actually took unstained white blood cells and looked at them under a microscope, they might look more off-white grayish like this, and even lighter than what I'm showing right here. They are not that natural pink and purple, even though when you see these blood smears, they're typically shown as a pink or purple color. Now, why do they appear pink and purple? It's due to this special type of, or actually not special, this is like if you go to a histology lab or a pathology lab or a cytotech lab, you will see this is like probably the most common types of microscope slide staining you'll see on human cells. It's called an H&E stain or a hematoxylin and eosin stain. And this is a very popular stain in histology. Chances are, if you look through your book and look at similar tissue images, most of the time it's some sort of H&E staining. So what is hematoxylin? Well, you don't need to know the exact chemical structure, but what does it do? Well, it stains things purple, but more specifically, it binds negatively charged molecules. So hematoxylin will stain things like DNA and RNA. DNA and RNA has a lot, they both have a lot of negative charges on them, their backbone, especially with that phosphate group. So phosphate has a negative charge, so the hematoxylin is going to stain this negative charge. So where do you find a lot of DNA in your cell? Well, where is the big warehouse of DNA? Your nucleus, right? So all these dark purple blobs you see here, these are the nuclei of these respective cells. Now eosin, instead of binding negatively charged molecules, it's going to bind positively charged molecules. And this is what we see, like a lot of proteins have overall positive charge, including hemoglobin. That's why we have our red blood cells, which are full of hemoglobin staining red. Now, notice that this special eosinophil, well, this is also why it's called eosinophil. Like, remember, phil, just like hydrophilic means it likes water, eosinophilic means that a cell likes to stain with eosin. So this is what we see with our eosinophil. Now again, this H&E stain is not how your cells look. Like your cells, if your cells look, you don't naturally look pink and purple like these cells right here. So what we have here is a tissue sample, and I believe it's a prostate tissue sample. And if you look really closely, maybe you can see the faint outline of the tissue here. But why do we use H&E staining? It's to make it easier to visualize. So that little slice of tissue is there, but once you add that hematoxylin, eosin, and other things used to help stain and get the dye into these cells and help the dye stick to these cells, then you finally get a slice that actually is colored this pink and purple based on where negative and positively charged particles are.